So I would like to welcome everyone on the third episode of Hort Forum that has a central topic, leveraging molecular markers in fruit tree breeding from promise to reality. We have the pleasure to have as central speaker Professor Ksenija Gasic from Clemson University, and also to have four panelists from the United States, Tom Graziel, from New Zealand, Richard Walsh, and from Europe, Marco Cirilli and Jose Cuero. We also have the pleasure to have Francois Lorenz, the president of ICHS, as moderator of this third episode, since it is tightly aligned with his area of expertise. So I will give the floor to Francois to moderate the webinar.
Thank you, George. Thank you. First, thank you very much for the organization of this series of forum. And thanks to Ted also. Ted is the vice president of ISHS, and George, you kindly help us to organize this kind of thing. Just to remind everybody that the spirit of this hot forum is not to have classical webinar, but to have to provide information, but also to provide exchange and discussion between the panelists, the speaker, the panelists, but also with you. So be ready to use the Q&A facilities to ask your question, and if the questions are not easy to understand, we can also give you the floor to ask your question. So the way we will organize so I will try to moderate these people. So we have one main, as George said, we have one main speaker, Dr. Ksenija Gasic, from the University of Clemson in the US, and she will make the introductory talk, and then we'll have the different panelists who will be able to discuss and speak. So I will give you Ksenija, so you are a professor on horticulture. You are also a breeder, well-known breeder and geneticist, mainly working on peach. You are dealing with all kind of traits, food quality, disease, and the good thing you are working on fresh fruit, but also on rootstock, which is I think one topic we also can put in the discussion, because it's I think not so many people, and it's I guess a very important support for many issues. So you have the floor for 20-25 minutes, and then we will open the discussion with the panelists. Can you share your screen?
Okay, thank you, Francois, for the introduction. Okay, just a second. Can you see my screen?
Perfect, thank you.
Okay, great. Okay, thank you very much for inviting me to give this talk. I was torn about creating it, so I hope I cover most of the things that people expect to hear. But as you see, okay, let me see if it progresses. It progresses nice. Okay, this is kind of the overview of the talk that I'll cover. I'll first, of course, introduce the Rosaceae family, and especially focus on the tree fruit crops, cover the resources that are available, and then discuss briefly about the DNA-informed breeding, and mostly showcase some of the rose breed accomplishments, which I call the rose breed effect. And then complete that with the case studies from peach, where I actually show you how I'm using this DNA information in my breeding program. So going to the Rosaceae family, we all know that Rosaceae family is very diverse, and that provides the nutritious food that contribute to our aesthetics, but also health and enjoyment. Those are all economically highly important crops, and they're related to the ancestral genome. On this slide, I'm just showing some of those that are important, and they're, okay, sorry, it moved, okay, moved to the wrong direction. And so, Do you see, do you hear the, okay, it went away, sorry. Okay, so some of these are berries, palm fruits, stone fruits, and they're all economically highly important. They're produced worldwide and mostly in temperate climates. Most of them are grown for their fruits, but some are in a fresh or processed form. So in this presentation, I will focus on those that are, of course, tree fruits. As I mentioned already, amazing diversity. They're highly heterozygous, and some are obligated outcrossers, which actually results in the great phenotypic and molecular diversity, which is explored in breeding. Why do we need so many different cultivars? Is because, as you know, environments change, and the growers or producers' needs change with the environmental changes. Also, the consumers' preferences are changing, and all of that needs to be addressed through providing new cultivars for the industry. One of the problems is that it takes time. So many of the things that people or industry or producers or consumers want now are actually 15 or 20 years in making if we don't start on time. So one of the key features for Rosaceae family is that they're very similar. And in 2009, the first effort started of producing the full whole genome sequences, and it started with apple, peach, and strawberry. So taking advantage of the sequence availability, the comparison between the genomes was performed, which is shown on this slide with the orthology map, which showed that shared ancestry I mentioned before and genetic similarities. Many of these are explored in modern breeding programs today, and some of this you could kind of see, you will see through next slides when I show them. So, from the Rosaceae family, of course, as I mentioned several times, what we'll focus on are the tree fruit crops. Why tree fruit crops? This table actually shows just the US production, and I took the information from the paper that we published at the end of the Rosebreed project. But just to emphasize how important these crops are, this table shows you the bearing acreage in hectares, total production in metric tons, and production in thousands of US dollars. So altogether, it's more than 10 billion worth production, and that was 2018, which I would suspect is more than that today. So there is an importance, but there are many challenges that the crop producers are facing. Some of these are listed here that are related to the productions, such as diseases, pest, yield security, post-harvest losses, labors issues. Then also on the side of the consumption, which is associated with flavor, texture, appearances, and for many of the rosaceous crops, having the consumer decline because of the quality of the fruit displayed on the shelf in stores, not actually related to the quality produced in the field, which is something that hopefully we can address in some other forums about the post-harvest handling. Also, the performances of the cultivars are often region-specific and product management-specific. And as we know, some of them are recognized as the cultivars and names on the market, but many of them are not. So within the Rosebreed project, what we try to sample and see is how many breeding programs do we have in the United States that breed rosaceous crops. And this table shows you what that surveying yielded into, and how many have a public, which is orange, and private, which is brownish color. programs in 2018. So about 100 programs, but many breeders actually breed several crops. So we only had 53 head breeders that we surveyed, but 100 actually breeding programs. So breeding is done traditionally. Even with all of the talks that will follow this slide, we have to rely on traditional breeding, and the DNA information that we want to use is just a supplemental part of that process. So in this slide is the feature of the apple breeding program and the timeline that's involved from the moment the cross is made to the moment that the cultivar is released. So a typical breeding strategy is a pedigree approach in which breeding populations are based on biparental crosses and they are generated based on the empirical experience, based on the phenotypes and impression of general combining abilities of the parents. In most cases, the heterozygosity existing within the rosaceous crop results in wide range of phenotypic variations that allow for selection of the traits that we want to combine in one individual. However, that means that the numbers is the game. We need high numbers of individuals to get that combination of traits that we are going after. Additional problem that is created is that that high heterozygosity also is coupled with low heritability and polygenetic control of many of the traits of importance that complicates breeders' ability to identify the best parents and best parental combinations. And that's also especially complicated in polyploids. The challenges also include the size, large plant size of the tree fruit crops, long juvenility that can go from 2 to 7 years. Then in addition to that, if we want to combine the disease tolerance or resistance with the fruit quality, and if we want to combine several diseases into the one individual, that also complicates the possibility of actually obtaining that type of a plant and extends the time needed, because traditional backcrossing does not work because of the high heterozygosity of the crops. So here I'm just showing the peach story that, again, all of these mentioned before applies to, where the process applied, particularly in my breeding program, goes to about 15 years before the trees or selections are moved into the replicated trials. So what are the resources available for breeders to make this process more efficient and more efficacious? So all of us here, I believe, know about the genome database for rosaceae. In 2009, the scientific advances accelerated building of resources for all crop species, but particularly for fruit trees or rosaceous crops. We started with the low density genetic linkage maps, then went through the apple, peach, and strawberry genomes, and then all of that data were housed in the genome database for rosaceae that was initiated in 2003. This is now the standard for rosaceous crops research. In this slide, you can see a list of some of the resources that are publicly available at the GDR. Also, some international community effort resulted on development of SNP-based genome scanning tools that provided genome-wide genotyping platforms. The value of this was that it allowed comparison of results across various breeding programs, not only within US, but across the world, because most of the breeding programs use these platforms to obtain the molecular information. So here are the cross-check of what's available right now in the GDR for the tree fruit crops within the Rosacea family. So we went from 3 genomes to almost 80 genomes or more, many maps. Oops, okay, I'm gonna have to be careful with my mouse. Markers in the thousands and millions, QTLs, which is quantitative trait loci information, the marker Mendelian trait loci or MTLs for different traits, genotypes, and mostly now or lately, rather say, SNP-based genotypes. We started with SSRs, but then moved with the SNPs based on these platforms availability, and moving into some more concrete information as the haplotypes and their phenotypic values. So one of the biggest issue during the rose breed, but I think not just associated with the rose breed, another big and influential project from Europe, Fruit Breedomics, also contributed to all of the information that I'm going to be showing through this presentation, is development of the standardized phenotypes, because they enable combining of data sets across programs for improved statistical power. And that will ensure that trait DNA association could be accurately determined. These phenotyping protocols are all available on GDR as well. So within the rose breed, what we developed also was the reference germplasm sets. Why that was important is because all of these information was released to the public through the GDR as well. And in this pie chart, you can see what is the number of germplasm information for phenotype, genotype available in GDR right now. This is remarkable number, knowing the pedigree connections between worldwide breeding programs that allows breeders to use the information generated in another program and potentially apply it in their own program. And I'll go back to that a bit in some of the examples that I'm using in my program, and we go to that part of the presentation. So DNA informed breeding, specifically we are talking about the marker-assisted parent selection or MAPS and marker-assisted seedling selection or MAS. And of course, as I mentioned in the first slide, a rose breed effect that I'm kind of trickling through these slides. So what do we mean under the DNA-informed breeding? So the goal of breeding programs is to be efficient in developing new cultivars, to make the right crosses that will generate the right combination of the traits in the progeny, and to focus only on those seedlings or hybrids that have a potential to have that combination of the traits. So DNA information can actually help us to choose the target, then to make the combination with the parents, right parents for the combination of traits we are going after, then to make effective crosses, progress this only seedlings that can have the combination of traits we are going after, then to trial the best selections, and of course end up with the main ultimate goal of releasing the new cultivars from the elite selections. So basically, in short, DNA information supports breeding decisions. One of the things is, how is that actually done? Well, mentioning in the section slide, through the marker-assisted parent and seedling selection. What does it actually mean? Marker-assisted parental selection basically traditionally was done based on the phenotypic performance of these parents and what we know about them. Now, with overlay of the DNA information, we can actually determine large effect alleles in parents and create effective combination for crossing. For the marker-assisted seedling selection, what that means is that we can identify seedlings that carry desirable allelic combination in families. So here on the slide are some of the traits that were first targeted within the rose breed and also fruit breedomics projects for development of the marker test. Many of these traits were discovered prior to rose breed project being funded, but information that was readily available for breeders to actually use for marker-assisted parent and seedling selection was not developed. Some of the reasons why that was not the case is because we wanted to do high throughput phenotyping or genotyping, and that was actually a logistical nightmare. And because we wanted to do thousands of seedlings in a short time, markers would provide that ability. Some of the logistical challenges for marker-assisted seedling selection is actually implementation. And here you can see examples of how it was done in the apple breeding program. Later on, I'll show you some of the modifications I am doing in my breeding program. One of the key problems for implementation of the DNA test available was the expense needed for the DNA extraction. Why I'm saying that is because at the start of when you make the crosses, we all want to achieve the high number of seedlings. When you have 10, 15,000 seedlings, like usually and normally you achieve in the apple breeding program, that's a lot of money to throw away on just the DNA extraction if you're going to retain, excuse me, 10 to 20% of material. So that was some of the most effective improvements were to some of these logistical issues to implement marker-assisted selection with either with the parents or with the seedlings. So an additional level of complexity becomes when the traits are not controlled by the few number of genes, but many genes across the whole genomes. So within the rose breed, we tried to do the proof of concept of genome-wide prediction and selection, and especially trying to look into the global genomic prediction, which is something that rarely any breeding program can do. There are many reasons why some of the boundaries of moving the plants across the borders, and especially me being in Prunus because of the virus issues and stuff like that. So you are not able to test your material in different regions around the world. So some of the things that could be predicted from the genetic information is the how some of the material performed and evaluated in one region can actually perform in another region. We did some case studies with apple and peach and showed that predictions go up to 80% of the performance in the different regions. This is, of course, thanks to all of the breeding programs around the world, majorly Europe and US, that contributed their phenotypic data and use those genome-wide SNP platforms to genotype their material that allow the cross-compatibility and use of this global genomic prediction approach. So this table and several slides show other tables split into the traits that at the end of the rose breed, we assembled information, okay, how many traits did we cover and how many of those discovered, which is in the blue color, validated in green color, and if it's underlined, that means that the DNA test is developed for this trait. So you can see that there are many fruit quality traits for apple, peach, sweet cherry, and then sour cherry, of course, didn't mention at the beginning, but the ploidy levels in the rosaceous crop is also complicating the genetics and discoveries. Most of the rosaceous tree fruit crops are diploid, but some very important ones are tetraploid, hexaploid, and for example, apple and pear are considered diploids, and they behave like diploids, but they are actually polyploids ancestrally. So there's also many layers of complexity to detect, validate, and actually develop the DNA test that would have a high predictability that breeders would trust. So this is for the disease resistance and phenology and productivity. So what we try to look at in within the rose breed is the how many breeders used the DNA information, and then found out that 73% of the programs actually used the DNA information by late 2019. And then here you can see the specific application where they mostly used it to detect identity of the material. Next level was for marker parental selection, and the least, of course, for marker assisted seedling selection and the upstream research. So we coined it as bridging the chasm between the available resources and actually application. So now we are moving into the peach case studies, and of course, the most available traits are shown on this slide that were available at the time we started. But some of these traits, you would argue that breeders would not care that much at the early stages of selection. And so in my program, of course, driven by the climate changes and the loss of production in the Southeast, we were focusing on chill requirement or some of the dormancy traits, and developed a DNA test which was based on SNPs that had a 70% prediction accuracy in the material. And this slide shows you the chill hours, and blue dots are actual phenotype, and yellow dots are actually the DNA test prediction. The next one is basically production and the fruit quality, which is ripening date and soluble solids. And we actually achieved a very high predictability and accuracy specifically for the ripening date. Slightly less for soluble solids, but that's also understandable, because soluble solids are highly dependent on environmental and on the sampling, which goes back to the phenotyping protocols you use. So among the disease tolerance, the Rosebreed 2 project focused on the 2 diseases per crop, and 2 major ones were bacterial spot for peach. And in this one, we were focusing first on the fruit disease tolerance, and we developed the test, DNA test, with a high accuracy, combining with the DNA extraction developed for strawberry that cut the cost on less than $1 for the whole test to be performed. So that really increased the use of this test, and this is the major one that I use in my program to cut the number of seedlings. Leaf tolerance was not as easy because it was polygenic, it's complex genetics, needed genomic predictions, and we looked into the genomic prediction, but markers as a random effects did not produce the high predictability of 0.2. And we wanted to compare the lab and field test. And then when we use the significant markers and as fixed effects, we actually increase the predictability. Similar story with the brown rot. It's also polygenic across the whole genome. So genomic prediction is the only way to deal with this disease. And what I'm majorly using in my program is actually to select the parents to make the crosses, because the high cost of the need to genotype the seedlings. And you can see here many of these different models perform the same. And what we majorly focused is to use the lab data to predict the field performance. And you can see in that graph on the right side how lab data predicted field performance. Accurately low or high field disease index was predicted, which actually gave us confidence that we can bring the fruit into the lab, in fact, evaluate the performance and use that data to advise for the selection of the parents. One of the things that I mentioned before for these germplasm sets that are available, because especially for peach, such a close connection between the pedigrees of many breeding programs in around the world means that any data that I generated for genomic prediction of brown rot in my program could be readily applicable in another programs, just without even sharing the knowledge about the material, but using it as a reference material or training population. So I hope I didn't cut too much of the time. Acknowledgement, of course, mentioned through the Roseberry Project and for the Clemson particularly. And I hope I raised your interest to pose the questions and ask the questions about actually validity and application of these markers in the breeding program. So with that, thank you for your attention, and I'm going to be stopping the share so we can actually.
Thank you very much, Ksenija. You're on time. Very informative talk. And you really consider the public we have, which is, I saw the list, so there are very specialists and breeding and geneticists, but also some people perhaps who don't know. So my request for all of the audience is to ask your question on the Q&A box, and so we will answer the question afterwards. And I will now give the floor to each of the 4 panelists. Perhaps start, since we are speaking about peach, we'll stay on peach and ask Marco Cirilli, for example. So you are from the University of Milano. You are quite young, but with already a long experience on different kind of crops, apple, peach, perhaps others, dealing with fruit quality, dealing with fruit disease also. So please, in a few minutes, you can introduce yourself, present your program, and the question that molecular markers raise for you.
Thank you, Francois. As a University of Milan, we are involved for many years in peach and apricot breeding program. The name of the breeding program is MASPES. With the name is not casual, because we were oriented to the use of molecular tool to assist the selection. As Ksenija, during the last 5 years, our main goal is to introduce routine application of a marker-assisted selection with the same logistic problem that Ksenija has already told. But the most important things that we face is what should we call the definition of killer traits. Okay, we use mass to select traits or seedlings, but for which traits the choice was rather simple in apricot, since shark resistance and floral self-compatibility are now the gold standard. But in peach, to say the truth, we are still looking for such killer trait, even if we have molecular markers for many traits. Maybe we are interested in a texture, and particular to the stonier traits, but in general we are still defining what are the priority traits to select with marker seed selection. So I want to directly go to my first question, that is an observation, that is our first criterion is it must be economically advantage. So the economic criteria for us is very important. It's simple to say, but often hard to establish because the literature is full of various claims, announcements about the economically advantages of marker seed selection. But I say it's quite poor on measuring the performance and their actual routine use. So the first question is, we would enter better in the economic sustainability of this approach and maybe establishing better whether they are convenient or not. What do you think about it?
Thank you very much, Marco. We keep the question, so and carry on filling the Q&A box.
Okay.
And then we will move to I keep the question about economic sustainability, which is I think a very
for me is a primary criterion and is good things to discuss.
Ksenija already spoke a little bit about that on the efficiency, and the Rosebreed project actually was also focused on that. Tom, if we stay on the stone aspect, so Tom Gradiel, you are working at Davis University in California, working on almond and peach, and also a geneticist and breeder. Can you tell us a little bit what you are doing? What are your main focus, and how you use the molecular markers, and what question you have, or what issues you have with them?
Certainly, again, Tom Graziel, University of California at Davis. I'm a professor here, but also a geneticist, plant breeder, as I often say, plant breeder, geneticist. My crops I work with are almond and processing peach, and we don't have much private breeding on those. And so it basically falls on the university to develop the varieties for these changing needs and changing times. And I want to really congratulate Xenia for a really excellent presentation on molecular tools. I think we're at a time where these very exciting on the leveraging of these tools, and particularly exciting because we're finally getting the tools to address probably the most important barrier to breeding progress, and that's consistently generating large breeding population sizes. Xenia mentioned that sizes of 10 to 15,000 for a traditional breeding, an applied breeding program to make the progress. We're dealing with killer genes, but for a variety to be successful, we have to understand that it needs no deficiencies in any of those multiple characteristics that the industry demands. So we're talking 20, 30, 40 or more traits. In order to achieve that type of selection efficiency, even with these molecular tools, we're going to need population sizes of 10,000 to 20,000 or more. The challenges with that has been both generating these hybrids and also once we have the hybrid seed, getting that seed to the seedling stage without the huge losses we typically observe. And in the almond breeding program, where we have a week to make pollinations, we've got crossing compatibility, we typically have rain periods. We've been able to achieve that hybridization success by using cage trees with bumblebee pollinators. I can consistently make 30, 40,000 crosses, even in these seasons, which are really challenging. The remaining challenge has been getting those seeds to maturity and getting them to germinate in high percentages. And again, we're seeing some really powerful tools. And again, Xenia's at the leadership of that, following up on some of Rodrigo and Fonte's work, using growth regulators to germinate immature seeds. By germinating immature seeds, we can go in, instead of going in August, where I'm battling birds and squirrels and diseases for getting those mature nuts off the tree and into the breeding, into the seedling stage, or the put them in the cold storage and worrying about loss there. We go in July. Once these seeds are physiologically filled, mature, we harvest those seeds in bulk, 1,000 at a time as we need, put them through the growth regulator, which is a GABA 100 parts per million treatment, built like so an almond within a week, we've got these seeds germinating. We can get them rapidly out into the field, saving us a year as well. So the barriers to these major limitations, population sizes, I think are really coming down. If we really want to make progress with fruit tree breeding for a lot of these programs, this is the major barrier to progress. We need to look at each individual barrier. So within the seed germination, it's not only seed germination, stratification, but loss from birds, loss from rodents, loss from disease. We can avoid these in a systematic sort of way. So very, very exciting times. And thank you for the opportunity to give my presentation and my views here.
Thank you, Tom. And we will come back to you during the discussion, of course. Now, what about cherry? Jose Piero Garcia is a colleague from INRAE working in Bordeaux on cherry, and with straight many links with fruit quality and I would say impact of climate change. Jose, you fine?
Yes, thank you, Francois. So I work as a geneticist and breeder at INRA since more than 15 years now on sweet cherry. And yeah, we're also using molecular markers since a few years. But in cherry, one of the problems that we have is that many traits are quite complex. So we have some single traits like Ksenija was showing, like fruit color or cell fertility, but other traits that are highly impacted by environment can be very complex, like, for example, the tolerance to rain-induced cracking, which is a very important problem in sweet cherry. And also one of the major issues is, as Thomas said, is the difficulty in sweet cherry to produce large populations. We've also used cages, bumblebees, and so on. But we have seen that some combinations are really difficult or for some of them, it's really difficult to produce large numbers. So we have to be creative sometimes. Maybe we can try to combine different parents together and see which ones work better. We can also work with open pollinations. I think molecular markers can also provide nice information there. And because definitely if we want to combine a lot of markers for a lot of traits, it's good to have large sizes of populations. If not, it's not so convenient. But in terms of economy, to link with what Marco was saying, I think that when you can eliminate hybrids that for sure are not going to be interesting for you, because they will probably don't reach, for example, in the case of sweet cherry, size of fruit, which is important. This is going to be interesting because when you plant those hybrids, they will remain many years in the field. So in the end, I think that even with the problem of the costs of DNA extraction and so on, today some of these markers are already quite interesting in terms of economic gain.
Did you calculate the economic gain?
Well, yeah, I did some calculation. I don't have them in mind now, but yeah, for sure. When in our case, when we consider the cost of the maintenance of the plots per year, and knowing that we're going to keep these hybrids at least for 8 years, because cherry juvenility, the cherry trees have a juvenility period of 5 years in our conditions. For sure, it's profitable. Of course, there is always the risk of losing some hybrids that finally might not be so bad, because the markers that we use for fruit weight have not, let's say, a predictive power which is super high, but let's say it's high enough to be quite confident that these hybrids we eliminate will not produce very big fruit, for example.
Thank you, Jose. And the
welcome.
best for the end, at least the one who is still should sleep at this time, Richard Volz is coming from New Zealand. And New Zealand, it's what time is it? 6 or 5?
5:45, yeah.
5:45. So
yeah.
And Richard is a breeder in plant and food for more than 20 years now, Richard.
Correct.
And with the same kind of objectives, and also using quite a lot of molecular markers for a long time now. So please, could you share your experience?
Sure. Yes, so thank you for the organisers for inviting me onto the panel. I look forward to the next hour and a bit of discussion. So yeah, as Francois said, I've been involved in apple breeding, cultivar breeding, apple and pear cultivar breeding actually for over 20 years at Plant and Food Research in New Zealand, as well as being involved in some other breeding programs in other countries. I'm very interested in the application of molecular markers in breeding programs, although I have been involved in some other research programs with the aim of developing markers. But as I said, my main interest is in the application. And we've been applying markers in apple for, I suppose, over 15 years now, both at the terms of marker-assisted parental selection and marker-assisted seedling selection. What we've noticed over time is that, as the previous panelists many of the previous panelists have said, there are a large number of traits, over 40, 50 traits that we need to be considering in apple, around fruit quality particularly. But what's happened over time is that we've ended up stacking additional traits, particularly around disease resistance, into those fruit quality traits to be considered as well. So the number of traits is increasing all the time. And as previously said, in order to develop a cultivar with all those combinations of traits at the level that's required, we need to have large populations. And the most efficient, and so we're dealing now today with over 70,000 seed and seedling, well, just a bit less than 70,000 seedlings to generate the 1 or 2 cultivars that might come out of those sort of populations. And in order to be efficient, we need to be applying markers where we can as early as possible in the cycle. In other words, in the glass house, straight after germination, to eliminate those seedlings that don't have the traits, particularly around disease resistance, that's required. And fortunately, in some ways, the genes that the markers that we're using are to do with major genes and scab, and particularly with apple scab resistance. And the whole idea there is to stack genes together to make a more durable type of resistance. And we need molecular markers to be able to do that. And so, as I say, most of the molecular work, the marker work, is actually around major gene resistance to apple scab. So I suppose if we look at most of the other traits, as previously said, most of them are quantitatively inherited, polygenically controlled. And for us, while we use some markers for choosing parents, we don't apply individual markers for many of those traits because they aren't very effective, very predictive, and therefore the genomic selection technique tool is very attractive. We've had some work early on, maybe 10 years ago, working on genomic selection. Initially was interesting, but then wasn't very useful in terms of predicting in the selection population, the generation on from the training population. So I suppose while it's a very interesting technique, this genomic selection would be very useful for us. I suppose the promise of genomic selection hasn't come through, and I'd be interested to hear what other people have to say about their experiences with genomic selection, because obviously it's been used in many other species, particularly animal. Can we learn something from the people today about their experiences and what's for tomorrow around genomic selection? I'll leave it at that.
Thank you, Richard. My proposal is to so I keep the main topics you raised: the cost, the problem of quantitative traits, genome-wide selection, the size of the population. You didn't raise at all the problem of phenotyping. I think phenotyping large population means a lot of time. So perhaps even if the topic is focused on the molecular markers, I think when you develop molecular markers, you need to have this phase of phenotyping first. So we can go back on all these points. I will just go on the questions. So I saw, ksenija, you already answered most of them. So the very technical one, so for those who want to look at the ksenija answers, you can go on the Q&A and that. So I will not, I think, not enter into the detail of the very technical aspects about the Illumina availability. I think one question from an anonymous person, how do you validate the markers before implementing in your program? So perhaps you can just explain what you said and ask if the other panelists want to add something. It could be interesting to share your experience in few words.
Yeah, I mean, before I actually answer that question, I could kind of cover some of the questions posed by the panelists, what I actually do in my breeding program. How do I decide what to use? So for me, the disease is the main issue for the growers, and my breeding program is placed in the environment that not often does have the conditions to show me the disease performance on the hybrids, which is a problem. So that's why my focus was first, okay, let's try to develop something to help me eliminate the material that's not important, because it takes at least 4 years or 4 to 5 years to grow the tree in the field before you can actually see the fruit. And then, you know, and so if I don't see the disease performance, I can actually advance something that in the end would not be improvement. We have many cultivars that are released annually for peach, 40, 60 biannual are actually registered in United States. But one of the problems is when you move them to different locations from where they were bred, they actually fail miserably because of the environment differences. So that's why I use the key diseases for us here are the bacterial spot for fruit and the brown rot, of course. So we got lucky with the bacterial spot because there were 2 genes that are majorly controlling the tolerance and the DNA test that developed. So how do I validate it? Basically, the first thing is using all of the information generated by the rose breed. We look into the haplotypes and associate with them the phenotypes that we combined. Then when we focus on the SNP that would predict each of these haplotypes for which we know the phenotyping performance, then we assemble the a set of 86 cultivars from that are important for peach growers, and run the test on them to see if the prediction matches the phenotype, and then expand that to my seedlings in my program, because we annually phenotype them for all of the traits. And as you know, breeding program has seedlings in various stages, so we apply it to the earliest one to the latest, to the point that I now generate about 4,000 to 5,000 seedlings, but only advance less than 1,000 every season, because I use the marker for bacterial spot fruit prediction and eliminate anything that's not tolerant and based on the marker.
Thank you, Ksenija. Are there other answer to this interesting question? How do you validate?
In our experience, we are more stringent, and we only use candidate variant specific markers. So variant that were tightly associated to the traits. For example, in case of brown rot, that is a complex traits, we definitely move to other approach. We are trying to implement a high throughput phenotyping approach, because we think that in the medium short term, marker synthetic selection for this trait would not be feasible. So in this case, these tools was deemed not sufficient of efficient.
You mean for this target, you prefer
Yeah,
to
for the
develop
target.
phenotyping tools than
Yeah,
but
of course.
actually it was the aim of my question before to
Yeah.
see how you balance between marker selection, genome-wide selection, or and phenotypic selection.
For sure, is a tool. All is a tools, not a an objective. Clear.
Any other comments?
Yeah.
Tom?
Yeah.
I saw
Richard,
you moving. Sorry, Jose.
no, Richard, I think wanted first, and then I can complete.
Richard,
Go ahead.
okay.
Okay, yeah. No, validation of markers is very important. We spend quite a lot of time validating markers, and we're talking about particularly the high throughput markers that we're going to be using in our marker-assisted, particularly our marker-assisted seedling selection, but also parental selection. We have a set of over 100 germplasms that we test in our elite material that we're using as potential parents. We also validate every parent that we use in our breeding program for a particular trait. We would every year we would validate the marker for that particular parent. And then in additional to that, we're also looking at, particularly if the marker is not a gene marker, in other words, it's a linkage marker, we're looking at possible recombination events. So it means basically retesting or testing the progeny to make sure that it has the gene that we are predicting from our marker. And if we've got good phenotyping techniques around scab resistance, which we have, we can do that. So quite a lot of work goes into validating the marker over the course of the work.
Any other comments on this specific question, Jose?
Yes,
I can make
Tom, sorry.
the importance of marker validation is crucial, but it also can create barriers to breeding progress. An example is seen with the almond breeding programs internationally. We've recently done a survey. Self-compatibility is a crucial gene for new varieties. The development of markers, validating markers, is very difficult because they're brought in from other species. From each species, you need to develop specific markers, and it's proven difficult for many of these species. The consequence is virtually all of the breeding programs in the world have used one source for self-compatibility. That's the Tuono variety from Italy. The exception has been UC Davis, where we've used multiple species. But that use of one source by breeding programs in France and Spain, Italy, Iran, Australia has resulted in tremendous inbreeding in these breeding populations because of that recurrent parent having that trait. And as a consequence, because almonds very vulnerable to inbreeding, a lot of these programs are seeing loss in yield potential.
Thank you, Tom. Jose?
Yes, just to complete that, to say that in INRA for sweet cherry, we also try to validate the markers not only on the elite cultivars or elite genitors, but also on larger collections of genetic resources to see if at certain point, if we want to do some more crosses more oriented in pre-breeding, if these markers are still going to be useful when we're going to use, for example, land races and so on. And also one way of validating, which is important, I think, for the private breeders that or private companies that can work with us, is to show the usefulness of these markers with phenotypic data. So, for example, in our case, when we've used the markers for fruit weight, for some crosses, we have kept individuals of each genotypic class. We have planted them, and in the last years, we are acquiring phenotypic data to show them that indeed these trees produced, in average, of course, fruit weight that was lower than the ones from the genotypic classes with the good alleles, because this is good, I think, for complex traits like fruit weight, for which we don't still have all the major QTLs. We might be working with one of them, which explains 30% of the variance, for example. So this was just about this question, François.
Thank you very much. I guess we cover the interesting with this panel is you cover different crops, different regions, and different way of doing. So I think it's interesting. I see another question you answer, ksenija, but I think it's interesting to raise it. is and perhaps to promote something, the software or programs you use to manage your marker data. So there are the BIMS, the Breeding Information Management System you mentioned, ksenija. I know that there's one webinar sometime. I saw that it already happened or it will happen. So if you are interested in it, go for it. I had the opportunity to have a demo on that at the PAG or something like that. It was a few years ago already, interesting. So are there, for the others, do you have, how do you do? How do you manage your data? How do you use your data? If you don't, you don't. We just go fast. You raise your hand if you want to answer. If you don't answer, that means you don't manage your data. Richard,
Yeah,
no?
well,
Yeah.
no, I'll say something. Most of our data is managed still in Excel, to be honest.
Okay.
We work with another with a commercial marker company, and at the moment it's Excel, and so I'd be very interested in other softwares that would be better than Excel, because obviously Excel has some limitations.
Who is also doing using Excel? Raise your hand. Yes.
Actually, database is a very big problem for us.
Yes, but you know, database. I have some experience coordinating EU big projects. Every time the problem is the database, and even to tell you the truth, even internally it's difficult. So we also use Excel files.
Excel,
and we are not happy, but it's fine. And we work on the database for 20 years or 30 years, so it's still ongoing. So thank for the question and thank for your answer. Perhaps ksenija, did you have experience on BIMs?
That's not to say that I'm not using Excel, but that's the start. Everything starts with Excel, but then I transfer it to BIMS, because what Excel doesn't give me is the quick way to see what I need. You know, so for example, I got asked by my grower, oh, do you have a combination of this trait ripening at this time and this and that? And says, okay. Prior to BIMS, it would be really hard for me to get that from my hybrids. Now, the breeding information management system within the GDR is for the rosaceae crops, but they also developed it outside of the rosaceous crops. So any program can play with it and test it. Of course, the problem is putting the data into the BIMS, you know, I mean, like with every database, you need to actually import your data in, but it's really valuable, especially when you combine the genotypic and phenotypic data. And what's mostly valuable for me, that it's connected to all the resources that are sitting in the GDR. They also made a connection with the green. So for those in the present, like a forum that don't know what that is, those are the information from the germplasm collections across the US. So you can easily search right now for the phenotypes, but very soon when we plug in the genotypic data, potentially for the genotypes for these accessions, and then maybe find the source of the tolerance or the trait that you're looking for. So that changed for me, and I was in a way lucky, because when I started my program, it coincided with the start of the Rosebreed effort. So it wasn't just me, it was the huge number of US and international people that worked towards the same goal that made me more efficient in what I was doing.
Thank you, ksenija. The last question you answered, ksenija, about the multi-species array. I think you answer it's in
Yes.
progress story. So, and perhaps a little bit more technical. So you give the answer.
Just to
So now,
say that Illumina arrays are no longer available. And so people are cognizant of that and trying to come up to the terms, what to develop that is most cost effective, because that's all for all of us. That's the first thing, how much it costs. per marker, per sample. And so David Chanier is part of it, whole world, kind of our world of researchers is involved, and we are working on it. So hopefully something very soon will come up that everybody agrees on.
Yeah. So I just come back on the general question. Nobody answered so far. So Dr. Aaron asked, so thanks for your presentation, Ksenija. And so it's a general question for you, and a very interesting question to open to non-temperate fruits. About subtropical fruits, what do you? How do you think from your experience from I would say all crops, apple, peach, and other are quite well known, and there are a lot of work on it. Do you think it's easy to transfer a part of the knowledge or the process from the discovery of the marker to the use, practical use in breeding? Do you have any idea to answer this question?
I cut those slides out of my presentation because of the time, but we are fortunate within the Rosaceae family that some things discovered in other crops are actually applied to other different crops. So what I'm trying to do within my program, I'm trying to use the mutant series from Japanese plum, from Santa Rosa family, to figure out what's going on with the softening of the flesh, because in the peach we do have the differences, but we don't have the mutants of the same genotype, just behaving differently. And so the Prunus family is really fascinating because they're so different, but they're so actually very close and can inform each other. Like between the sweet cherry and peach, many traits are just phenotype and genotype in sweet cherry or in peach. But when you cross-check the genomes and overlay it with the information known, you can find many connections and the same genes being involved. So as for the mango, I'm lacking knowledge about the mango as a crop and its genetic resources, but I believe that some simple cross-checks could give you at least the starting point to understand how close it is to some other species and how you can actually use something that's already developed for some other species to apply and test if it works in your crops.
Thank you very much. I tried to look at all the questions. They are very interesting questions. So I have in front of me the question of Morgan about genomic selection. We can keep it. Ksenija and Jose answer about the question about rootstock. I think we can spend a few minutes on rootstock and then move on the genome-wide selection. So of course it's a key topic. Jose, perhaps you can, or Ksenija, you can just comment what you said, just as an introduction, and then open the discussion.
Yeah, I agree with Jose's statement, and I was hired to be the fresh market peach breeder. So no rootstock, no processing. But situation in the field changes, and growers were adamant that they needed help because we are having multiple problems in the fields, and the soil and the rootstocks that we have do not address those. And the major things that what I saw was that there's lack of the resources for the rootstock breeders. And in addition to the timeline that we showed for the fresh market crops, fruit-oriented crops, rootstock is like twice the time, because you first need to develop rootstock characteristics and then test it in combination with the scion, which actually prolongs the time. And that should have I agree with, put more emphasis or actually directing the efforts to the rootstocks. But one of the problems, as you saw, we have like about 100 breeding programs for the rosaceous crops in the United States. I believe we have maybe 2 or 3 rootstock breeding programs, you know, and that's kind of.
It's the same in Europe. The number is decreasing in pig fruit. The number is decreasing. And in your place, Richard, you used to get apple rootstock breeding program 20 years ago.
Yeah, we still have a rootstock breeding program that is very active, and in fact, molecular markers are being used quite extensively in that program, particularly for pest and disease resistances. So, as well as some dwarfing characteristics. So, yeah, rootstock breeding is important. It's a lot longer term project compared with scion, and that places some challenges around, you know, it's financial from a financial perspective, but very important, particularly when we're talking about climate change and adaptation to, you know, environments.
Yeah, what the stress.
Yeah, I could like to make with rootstocks. When you look at prunus, the great majority of new rootstocks are species hybrids. And when you get into species hybrids, again, you get the challenge of it may be the same gene involved. First, you're limited to working with major genes primarily. Second, you really need to start to understand that invigoration from interspecies crosses, similar to hybrid vigor, but from at a species level. And finally, although you may have the same genes for nematode resistance, the markers development for those different species, say peach or almond or apricot, is going to be different, which is a challenge for us.
The problem is the length of the cycle of selection and the cost of the program. So we are at the European level for apple and pear. We are thinking of a sort of platform or collective to answer this particular problem of rootstock. So the idea would be to merge the few rootstock breeding program which are left, and to try to have one common program together. So it's something we are thinking, and because you know, so in Europe you have a lot of breeding apple and pear breeding program everywhere. Very few fresh fruit, very few for rootstock. So the idea would be to merge some teams to try to have a collective effort on that. Okay, so we all agree that rootstock is important. Good to know that Richard, you are carrying the effort on that, and waiting for your last selection. So now we move on the genome-wide selection, genome-wide selection aspects. So Richard, you raised the question. You in New Zealand have, I think, in Apple the longest experience on that, and you summarize a little bit your experience before. I saw on the list of the participants that there are also people who have experience on that, including my colleagues from INRAE. So let's exchange about that. What do you think it's a good? Is still an opportunity for the future? When and what are the bottlenecks? I think we know a little bit the bottlenecks about the price, for example. But what? So, Richard, I think you express you are mitigated on that. So so enthusiastic now. Just to
Sorry, what
you are not
what was
so enthusiastic on.
I think after the initial results, we were very enthusiastic. But just that it's, as I said before, just to repeat myself, that in the use of the essentially the genetic models in the selection populations and the follow-on generations, they haven't been so successful in prediction. But I think we've got to come back to the potential gain that can be made in terms of saving time. And I don't think we sort of need to just I mean, I'll just make the statement that saving time, in other words, we heard before, the traditional time from crossing to cultivar release might be in apple 20 odd years, but with the potential to actually save a significant amount of time through the application of genomic selection, we're getting cultivars into the, hopefully impactful cultivars, into the marketplace sooner. And that is a huge benefit, financial benefit to everyone involved, including the breeders, if they're getting some return on that, you know, on those cultivars released. So we have to remember the potential gain from successful application of genomic selection. So that's what we're working towards. The question is, yeah, how can we overcome some of the barriers that we've seen, and from a research perspective, try to pose the right question, pose the questions, and design the experiments to help us understand that.
I saw, so for the people who are in the audience, for example, Hélène, could you share your experience? Can you, George, is it possible to give the floor to people, or raise your hand and...
Yeah, we can give the floor. Tell me the name again, Francois.
Hélène, would you like to... I don't want to push.
Okay. No, I will take care of that. Ellen, you have access to the microphone and you can open your camera also.
Sorry, Hélène.
Do you see me? Yeah. Okay. Hi, everyone. Thank you for give me the opportunity to talk also about genome-wide selection. What was the question indeed?
The question is genome-wide selection a tool we could expect to use in the next year or in the future? Perhaps you can introduce yourself, Hélène. What is your background?
I'm a research scientist at INRAE. So I'm working in Angers, just not so far from Francois. I'm quantitative geneticist, and I try to work on the use of markers in breeding. So either marker-assisted selection or genome-wide selection. I've compiled some publication regarding genome-wide selection in Apple, and from this, what I've seen is, well, we have investigated accuracy of prediction for several traits and in various configuration of training population and validation population. And we have very various results. And so the first results that were obtained in New Zealand in the publication of Satish Kumar were, I think, almost never reproduced because they had chosen a very favorable situation where they predicted with full sieves. They predicted full sieves with full sieves from the same family, so it was really favorable. And that's not really the situation that we would have in real application of the genome-wide selection, because we want to advance and use new parents. So the seedlings that we want to predict are not full sieves from the individuals in the training population. And the other validations that have been done with real families from breeding program, I think in many cases there was not enough investment in phenotyping these individuals that were used to validate. So the inaccuracies were due both to the models of the genomic prediction and in the phenotyping that gave the validation data. So we have to invest more, probably, in validation. And probably we can also try to combine more data from various experiments. And also in Europe, we have built a reference population for apple that has been genotyped now for more than 5 years, I think. So we hope to obtain with that a really useful training population, but up to now, only in the Swiss programs they have taken this data to try to validate. But again, they perhaps do not have invest enough in phenotyping for validation. So they have only a few years, and they have a structure validation population that is perhaps not so favorable for good accuracy.
What you mentioned about the RefPop, Hélène, and so it's part of one EU project. We are all, a few of us here are participants. And the good thing, or the interesting thing with this project is it's a project which merge apple, tomato, but also maize, wheat, and things like that. And the main difference, so on maize and wheat, they made a lot of progress in genomic prediction. And you mentioned that, Hélène. I think one of the main reasons is that they have a lot of phenotypic data. There are a lot of phenotypic tools, a lot of high throughput phenotyping, which gave them a lot of very precise data, a lot of historical data too. So yes, I think the accuracy and the number of data is a key issue, I guess, for us. It's why why I mentioned phenotyping before, because we can't be efficient in using molecular markers if we are not efficient in phenotyping. You agree, Hélène?
Yeah.
Yes, thank you.
Thank
So,
you.
Jose, thank you very
Yes.
much, Hélène. Sorry for.
Yes, thank you, Hélène. Don't leave yet. No, I wanted to ask you, Hélène, and the others, maybe, what do you think about the problem of the cost of genotyping operations in genomic selection? Let's imagine the ideal situation in which in each crop we have already worked a lot on phenotyping, on nice design, as Richard was saying. So we know which parents we can use for our crosses. We know genomic selection is going to work. But what about the cost of genotyping? For example, have there been studies that have compared the power of prediction by varying in an important way the number of markers and the impact of this in the cost?
There have been perhaps a few studies regarding that. Particularly, I remember a PhD that I supervised was trying to compare genomic prediction accuracy with low density as obtained with a 20K array in apple, or high density obtained with a 480K, and they were almost similar. So we do not have that much diversity or that much generation of recombination, so that we would need to have a very high number of markers. So mid density could be enough, I think, for apples. I don't know for the other species, I must say. And regarding the cost, I think the multi-species arrays were suggested. In Apple, we are embarking in a project with, at INRAE, with other species that are that have much more means, just like wheat or maize or and so on, or seed rape. And so when we have a very large group, so with many individuals to genotype, with a multi-species array, we can try to have cost reductions.
Marco, thank you, Hélène.
Yeah. If I can just follow up with that, that's an excellent question, which we actually asked during the rose breed exercise with the genomic prediction. And one of the analysis showed that 500 informative markers are enough to reach the plateau of the prediction. So you don't need the thousands of millions of markers to do the genotyping. I can say that I did not test it in my program. We used about 1,500 markers for the genomic predictions that we've done. And but what we tried to develop was the subset of the markers that are highly informative within the material that was genotyped within the project. And we developed the with the LGC, the seq-SNP platform for the 3,000 markers. The same was done for strawberry and for peach. And we are yet to work on that, but that's on the list of the projects to complete, to test those markers to see how can we reproduce the predictability obtained with the full array markers for the genomic prediction. What I was told by the LGC is that once this initial cost was paid for the 3,000 markers, now breeders can actually use the subsets depending on the materials that they have, and genotype maybe only for 500 or 1,000, which would decrease the cost. The cost at the time was the $35 per SNP for all 3,000 markers per sample, and it included DNA extraction. So basically, you would just sample a tissue and send to them, and they'll send back the information. What we did, we tested this platform with the programs that participated in the rose breed and with several programs within the United States that wanted to test it. And recently I had a colleague coming to to visit me, and we went through his data and looked into the haplotypes that we recently defined for different traits, and we predicted in his material how they should perform for different chilling requirement on all of these traits that I showed that I'm using in my program, and they were highly accurate within his hybrid material. So one of the things that I didn't mention, we were always talking about, okay, we will save the money. I don't actually think the money is what's being saved. It's the time that and effort devoted to phenotyping, because traditional breeding will not be replaced. We all know that. But we want to just focus our attention on the worthy material. And so at the initial stages, maybe the cost is high because you need to genotype lots of materials. But when you compare that with the cost of growing the tree in the soil for 10 or more years, that cost gets on the lower level. So when we are trying to talk to within the university where I work, administration and everything, growers, we never say it's cheaper, so you get less money to do the job you're doing. That it's basically providing me with the possibility to more efficiently do my work and actually get more individuals at the end that meet the specifications at the beginning, the combination of the traits.
Yeah, the question is, and actually there was one question from Aurélien, but is there anybody here who already estimate the cost of molecular assisted breeding and or genome-wide selection? So ksenija, you presented some numbers, and Rosebreed did the work for the map. I don't think we are still we don't have numbers, really figures, yes, for the genome-wide selection. But the question, I think still have to keep in mind that the marker will not at one time we'll have to plant trees, and we'll have to yes, observe the trees. So the question is, when do we use at which step do we use markers assisted selection? At which step do we use a genome-wide selection? And the question you raise is it, do we save trees, or do we have that it allow us to have more individual at the beginning, so the same cost, but more power for the selection. So this is always the question. But of course, we will need trees in the trees on the field for a long time, because in our breeding program, for example, the first step of the selection is 5, 6, 7 years. So in 5, 6, 7 years, you killed I don't know 99% of the trees of the individual, and the rest, the 1% which stay, you need 10 years to select the last few ones, which is, so we have to be very careful about the what we save using the molecular markers, because at the end of course we'll need different testing everywhere by agronomists and by, we're not only planting a cultivar is not only the work of the geneticist of the breeder, is also the work of the growers, of the agronomists, and everybody. So it's I think it's important to take that into account. According to me, all the estimation which have been already made on the cost of the use of marker in selection have been very optimistic. I think we didn't do it very carefully here, but I'm not so optimistic about that. But Marco, sorry, you raised your hand for a long time.
Yeah, just to confirm what you say. For us, we have experience in developing a new approach based on single primary rickshaw technology in apricot. We design a probe for 25K SNPs, for example, but the cost is around 40, 45 euros. That is too much for any objective. So the price is actually one of the limits. The second limit is we have no sufficient knowledge of most important quantitative traits. This is the reality. Predictions are good, we publish that are good, but in the end, they are not so good to justify the cost that we are affording. So many of the traits that are interesting should be approached by multiple groups, very general project, because they are very difficult. Most of them are affected by management, and it cannot be easily phenotyping, because when we see phenotype one seedlings, mean that we have only one replicates. So we have to move on selection or germplasm. In case of the environment, we have to manage different collection, as you know, in apple or peach pop. So we need a lot of research before saying that this approach could be valid. In the end, if we consider the curve, I have in mind the human genome learning curve. No, in 20 years, the cost passing from 1 billion to less than 1,000. I hope that one time the cost of planting trees will increase, of course, because increase the labor cost, increase the cost of land, the raw material. So we will be a part of the curve where genomic prediction will be the most effective, but it's not now.
Of course, we have to think about the next future, not now, because I guess that the price will be
it's clear.
So I think we have to distinguish. Of course, we have to take into account the cost. But so now it's, I think, not feasible. But if we have also to see at which level, at which price, the genomic selection, for example, will be efficient.
Also, the knowledge has a cost.
Yeah,
the knowledge require a cost that is not so less.
Hélène, then Ted.
Yes, regarding obtaining trees, and I was just thinking about well, we need trees to be able to make crosses, but well, perhaps not trees that can be observed, because we could develop more protocols for having trees flowering in greenhouses and so on, to advance to have more speed in the process of crossing. And I think we should think about separating the cultivar development part where we need to observe trees and the pre-breeding or recurrent selection part where we want to raise the frequencies of favorable alleles. But for this, we do not need to observe the trees if we can trust the genome-wide prediction. So that was my point.
Thank you. Thank you, Hélène. But you know, the cost of a hybrid orchard is much, much less high than the cost of variety testing. Variety testing in many replication in hybrid, you can go quite straight. So I think the question of the cost is not so easy to wonder. Ted, so the pure geneticist.
So I have two questions. First of all, I'd like to know what the annual cost of the breeding program of Richards and Ksenijas and Jose's and Marcos, what their annual budget is for their breeding program that run this. I have a sense that we're talking mainly academics here. I don't know. I mean, I don't have the feeling that commercial plant breeders and tree crops are using that many of this technology or moving this forward very much. And I'm wondering if how much of this is actually practical in the commercial breeding business, because it strikes me that quite a bit of it requires a lot, not just money, but also technology and technical people who understand all this stuff. So just a few comments here, because if this only stays in the academic world, then I don't know how practical it is really.
So, Richard, you want to answer?
Yeah, I suppose that I mean, I could give you a number, which is sort of about 1.5 million New Zealand dollars for the testing, the breeding and the testing phase that Francois talked about. So I don't know if that means anything to other people, but I suppose I broach between the academic and the commercial breeding. Yeah, I agree with everything Ted said. Basically, we are applying markers. We do need quite a high level of technical expertise involved in deciding what markers, how the markers should be deployed, etc., etc. But I think, at least for our program, we have to use the markers for some of our scab-resistant gene stacking pyramided traits. There is no other alternative. So
yeah,
we're going to go down that path.
Yeah, I'm not against it. I'm just asking the question because
Ted, it depends, I think, according to the crops. That in one of the ksenija slides, she showed the public and the private program. So you saw that depending on the crops, it was figures from the US. But for example, in Europe, a lot of apple and peer breeding programs are not 100% academic, and there are also cooperation. For example, our program is an academic plus private program. So of course we take into account the economic aspects. And one of our aim and the deal we have, Jose, we are part of the same national cooperation with private breeders. The aim is to separate. It's what also Hélène said before. Separate what is the upstream part of the breeding, which is all the development of the markers and things like that, and the development of pre-breeding material to the commercial breeding, the way we are working. So it's very separate. And our aim is to transfer to the private breeders for example, the use of markers. Now markers, you don't do that by yourself. You pay NGC or other companies to do it for you. So it's just a matter of knowledge. And in our case, the private breeders are more and more. There was some in the list, but I can't see them anymore. But they are more and more trained and more skilled on genetics, on markers, And so now, at least at our level, the transfer is much more easy than it used to be 10 years ago. And ksenija showed the numbers in 2009 and the use of markers, and there's a big evolution. So I think all the project, Rosebreed, Food Breedomics, or the project help to do that. So it's also I think our duty as a public scientist to also transfer the technology and to be more focused on the more upstream part of the breeding. So we are not far from the economic aspects of TED. Tom, what is your experience on that?
Yeah, my experience is there is an interest within the private breeders to develop markers, but I'd like to use this segue as a caution. There's a lot of interest in private breeding, particularly for things like almond, because it can be very profitable. As Richard alluded, the breeding, the time to develop varieties is dependent on our selection efficiency, how quickly we can combine those genes that are required for a successful commercial variety. But the second component of that is regional testing. And Francois, you talked about the need for agronomists and growers. For a crop that I work with, such as processing peach and almond, this would be true with apples as well. To be commercially viable, it has to be productive over expected orchard life period of 20 years. That means it has to be compatible on whatever rootstock it is for 20 years, as an example. The only way to thoroughly test that, or to really validate that, is to have field testing for 20 years in all of the major growing regions that it's expected to be produced in. In terms of time to breeding new varieties, this has been the greatest barrier, which has been most difficult. It's also a barrier that I see a lot of private breeders ignoring, and we're seeing the consequence of that. Now we're seeing new varieties in almond, for instance, where we have millions of trees in the orchards, and just now at 10 years, we're starting to see that it's incompatible on a major rootstock. So that's a warning that should be particularly communicated to the private breeders.
Sure. And from one crop to the other, you spoke about the almond, which is very different from peach, I'm sure, and from other kind of crops. So difficult to have a general answer. Richard, you had something to answer to Ted's? No,
It was really coming back to the genomic selection chat. I don't know if we've left that. We've sort of moved on, maybe, but
let's carry on. So everybody, are you happy with the answers, Ted? Happy? I don't know, but
I'm not happy.
Ask one question.
You're not happy. Why?
Maybe I can add something to Ted.
Please, Jose, make Ted happy, please.
Yes. In sweet cherry, which is a difficult crop, with less advances as compared to apple or peach for many reasons that I'm not going to detail here. What I know is that some private breeders are at least using markers for parent selection, or to make sure that when combining the parents, they will get the highest percentage of superior individuals with those crosses. So far, this is the only use I've heard of for private breeders on sweet cherry. But some of them might want to use them for seedlings. But I agree with you, Ted, that it's a little bit difficult for them to move forward in this step.
Yeah. Also for peach and apricot is the same. All private breeders use molecular markers now, at least for in apricot for Sharka and self-compatibility, for peach for other traits depending to the objective. So it's rather common now also for private breeding programs.
Yeah, I think the self-compatibility was the first example of wide use of the molecular marker, even though if people did not maybe trust them that much, the issue is so big that they were willing to play with it and try.
Yeah. So my background, my involvement in breeding, I've been overseeing a prune breeding program for 40 years, and we've been struggling just to do the phenotyping to figure out what phenotype will actually go through the drying process. And prune being a hexaploid and a very difficult species to work with. And there's only a couple breeding programs in Prunus domestica worldwide. We tried to develop a self-compatibility marker, and but then it turned out it didn't work at all, and so we're back with nothing. But anyway, I'm just frustrated with the thing.
Actually, your point is it comes back to what the question about mango and things like that. I think we are here speaking about quite advanced species in terms of genetics. When you go in less advanced, and we see the difference, for example, between cherry, perhaps almond, and apple and peach. There are, I think, a gap in terms of tools, in terms of means. And so it's an excellent question to see how we also could transfer from one crop to the other the experience. For example, telling you don't use the markers for that, it's useless or things like that. So I think it's also the way we should collaborate together.
Well, I think there's a real difficulty in using in promoting all this marker assisted selection and so on in orphan crops or crops where there are very few people working. I mean, the promise of doing it, it's a huge leap to get into the kind of knowledge that Ksenija and Richard are talking about, you with the apple and so on, where you have hundreds of breeders and things. If somebody is on their own doing things in an orphan crop, it'd take a huge amount of work to get over that hurdle.
There's one comment on the Q&A from Dan. He said the major commercial use of these technologies are used in corn and some vegetable, of course, with profitable crops. Okay, yes, but I think on some of our species it can be profitable. At least it's not only a problem of cost. Richard mentioned that before. The same for us. The main use of the molecular markers so far in our program is to accumulate. I never can say pyramid to put together a different gene of resistance to make more sustainable resistance. So this is not a problem of this is something we couldn't do without the markers. So I think we are also to distinguish that. For me, for example, using the marker to select we have good markers for texture, for example, or for acidity. Is it useful to cull individual for markers for one marker on one trait linked to texture, taking into account that texture is a so complex trait? There are a lot of questions, but to accumulate a gene of resistance, of course, it's something we can't. We have to use the markers. So I think we have to also to balance. Okay, we answer your question, Ted. You are a little bit more happy?
Yep. No, but that's okay.
No. Tomorrow.
What I can actually tell you is when we had the peach conference in Greece last year, I was approached by several people that are from the industry sector or the private sector, telling me that they are actually using the DNA tests we developed for bacterial spot. So it depends on the issue and the problem. And if you are able to see it in the field, that's what forced me to work on those, because I was not seeing it in the field, because where I breed my peaches is far away from where the growers are growing them. And, and stuff that we bring from private breeding programs from California looks great there. And then it comes to the humid environment here in South Carolina and all hell breaks loose, And so some of these things justify the effort, and maybe even accepting the fact that you will probably lose something by applying the markers. But for me, going from 10,000 trees to evaluate in the field to 1,000, it's a big, big benefit.
The
Yeah,
problem
well,
is
we're going back to what Marco's first comment was, finding killer traits.
Yeah, exactly.
and finding the good markers, because if you cull half of your seedlings and you cull for not the good traits, it's not so good. There's one comment from Aurélien here discussing with private French breeders about the genomic selection. They are very scared about, yes, but if we discard individual which are interesting. So
but it's good.
Also,
it's good to work less. Huh? Anyway, it's good. We work less at least.
You are right, and actually, it's what happened with the animals. I'm not
and we
sure.
say, okay, it's predictive, even if it's not, but we work less.
Okay.
One of the problems is the marker is predictive, but I have my advanced selections planted now. I'm in the last stage in different environments, and well, the marker, and in South Carolina, they never got the disease, but in some other states, they are. And so what's going on? You know, so the marker is predictive for the race that I have here, you know, not for the one. So there's more work to be done.
Yes, and the question about decreasing the number is also a big question. If you decrease too much, you have less diversity, and so less individual to select for quality. For example, if you select for resistance, at the end you end up with few individuals, and you can't select for quality. So I think it's we have to balance and yeah. Okay, thank you. How much? Well, we have less than 10 minutes, so I will go through the other questions. So there is one question about the use of transcriptomic. So Joshua asked the question. I'm interested to hear if transcriptomic have been used in marker development in your project. I think yes, at least to validate the function of the gene, so on some traits. Considering traits that are influenced by environmental factors such as food quality, will RNA sequencing experiment that uncover gene co-expression networks and co-regularity networks lead to improvements in genomic prediction for polygenic traits? Là, you have 10 minutes, 2 minutes, 1 minute to answer.
Okay, I begin. No,
yes.
For me, it's no. We need no very experimental plan, very serious agronomic experimental plan, not transcriptomic.
Yes. We can use transcriptomic, but not only transcriptomic.
After genetic knowledge, first agronomic, then genetics, then transcriptomic. This my opinion.
Any other opinion on this point? Yes. Richard? No. Jose? Tom? No. Jose Chaparro, in our pitch breeding program, we have decided to use markers to determine the degree of difficulty for improving traits. Okay. Our generation time is less than 2.5 years, and so time resource saving are low. So any raise your question, comments? So it's, I think, really specific to peach because the time is quite different from other crops. No comments?
Yeah, it's actually our problems. The time is relatively short, so the feasibility, the economic feasibility became more shorter. We have to be very effective because the costs are lesser.
Yes, you are right, but
it's a problem actually.
But our problem in apple and cherry, I think, it's long and we don't have, but we have too many traits. So
but in peach is short, so the cost effectiveness should be great.
Yeah.
But on the other hand, you have a lot of monogenic traits in peach, for which the predictiveness is 100%. So depending on what you're looking for, it can still be useful, I think, to use markers.
Yeah.
No?
Yeah, but they are not so interesting. Yeah,
I agree.
I think mainly now with the climate change and issues we have, and there's insufficient chilling, and you need to actually, in my program, I try to breed between 600 and 800 chill hours requirement. And now I'm actually lowering down to 500 to maybe 700 because we don't get enough chilling, which is not case today. It's very cold. You know, so that's why we looked into developing these markers. And although there are several of them across several chromosomes, we focused on the major one on chromosome 1, just to get some information on the material I have and eliminate those that are low chillers and especially the high chillers, because I mean, we can no longer grow them here.
But Ksenija, this is interesting, but for chilling requirements, maybe there is a good correlation with bud break date of 1-year-old plant seedlings, and or not. I mean, this is the question, but I know that some private breeders in sweet cherry have been trying to use this as a proxy. In general, there can be good correlation between bloom date and chilling requirements. So I don't know for bud break at the very young stage, but
Can you just say yes? Tom and Marco say no.
One of the things is that I realized that chilling is not the only part of the story, because you know, heat requirement plays a big part in when the trees will bloom. So serendipity in my program ended up with the material that has about 500 chill hour requirement, but actually blooms the latest of all, you know. And I thought, oh shoot, this one is the high chilling because it's the latest blooming, but it's actually, when we did forcing, it's the, so you know, it's all above and all combined, you know. I mean, it's hard.
I hope, Ksenija, that we can provide you the information, because we have the results from chilling requirement of the RefPop sites. We have Murcia, Greece, and we see these. For example, that we have a region where it's dominated by chilling requirement, region it is dominated by heat requirement, and we hope to develop your model. For us, we are in a good environment, so we are not interested in. But for example, in Murcia, they're working a lot on this, mainly based on phenotyping. They have a very tight phenotyping schedule, and they don't use markers, but they are interested in, of course. So
thank you very much. So I think we answer most of the questions. We are at 3 minutes before the end. So I would ask you, each of you, to give a few seconds, few words of conclusion. What are the main message you can pass on? Ksenija, ladies first.
Okay, so personally, I don't think I would be doing what I'm doing now if it wasn't for the rose breed project, fruit breedomics projects. You know, when many people are needed for the initial efforts. Now that we have the resources developed, now I am able to go and target the traits that are interested for me or my growers, and to look more into them. Also, I agree with Marco about the killer traits. For me, it's the disease. Because if we cannot protect them in the field and you grow susceptible cultivar, there goes your production when the environment is favorable. So that's one of the key thing that growers said, we want the fruit, you know, tolerant to the diseases. And so then that cuts the number of individuals that you need to evaluate. So maybe some other easier and 100% predictable traits I could do, or I don't really mind if it's peach or nectarine, you know, if it's yellow or white and stuff like that. So that's from the peach perspective. As for the genomic prediction, right now I'm just using it to predict the parents and to make the crosses, and then I look into in the field and evaluate it like that.
Thank you, Ksenija. Tom, I just on the order of my screen.
Yeah, I'd like to follow up on Ksenija's comment that it is a team effort, and my major comment I want to make here is the efforts, particularly by rose breed, has emphasized not only the development of new information, but the development of toolkits that breeders, both with a good background into genomics and even those that don't, can start applying these things to improve their breeding efforts. So the Rose Breed, the Rose Exec, the Breedomics, very, very important tools. If you're not familiar with them, look at Xenia's PowerPoint, and she has directions to get those websites.
Thank you, Tom. Marco?
Yeah, I speaking a lot, so
No,
I have I have just a provocation, but given that I see the tendency to a private component starting to prevail in our public academy, I question to myself where this knowledge will be of public domain in the future. Yeah,
Okay.
it's the real questions.
It's for next Hort forum.
Yeah.
Thank you. Jose.
Well, in my case, I would just say that for the moment, we are trying to optimize the classical marker-assisted selection program with a more and more multi-trait approach. And we are still far from using in routine genomic selection. But I think we're going at least to try to explore the possibility. And also we're going to combine that with phenomic selection that we did not talk much about it today, but maybe it will be another topic for another Hort forum, hopefully. François.
Thank you, Jose. Richard, still awake?
Still awake, yeah, still awake. I suppose, yeah, as I said before, we're applying marker-assisted selection in our program, but it's interesting how, if I look at, say the last 20, 25 years, how much investment has been made in genomic technology versus phenotyping technology. If we take one of the key traits for us in Apple, which is juiciness, we're still doing the same thing as we were when I first started, sensory evaluation of juiciness, and that is quite not that precise. And we haven't really made any progress in terms of high throughput, particularly around texture in Apple. And I think in some ways that's limiting our ability to, for this very important trait, for us to make progress in applying some of these, or even developing some of the genomic tools that we need to. So I suppose a plea for a bit of work around, or quite a bit of work around plant biology, going back to plant biology, understanding and developing new tools for phenotyping.
Thank you, Richard. I actually prepared some words for conclusion. It was one of my conclusion. The last word, I think, for me, it's molecular markers, phenotyping, and things like that are only tools, and I think we still have to keep in mind that breeding is, some people say it's an art, and I really think it's an art, and the mind of the breeder is something you can't modelize, you can't change with molecular markers. So we have also to take into account the complexity of the work and the importance of the people who are looking, who are selecting, and who are thinking everything. So I think the role of the breeder is to use the good tools, but the tools will not do the whole breeding. So I think it's something, a good balance to have, and to really promote. We have some problems in our institute to hire new breeders, for example, which is a very different job as a geneticist or physiologist. And we really need to be powerful to hire new breeders, and which will be able to use the good tools. Thank you very much, everybody, for, I guess, this, at least for me, it was very interesting. I hope all the people who attended were also happy about it. Thank very, very much again to George and Ted for the organizing this webinars. I think we reached the idea of discussing, disagree, agree about the different points. And we cover quite a good number of hot topics on that. So thank you very much. And Have a good day, a good night, a good whatever, and see you soon. Bye-bye.
Thank you very much
Bye.
for moderating
Bye.
this webinar. Special thanks to Richard for the special hours in New Zealand. And also, I think that if the persons who attend this webinar are interested in a special topic, they can communicate that with ideas. I think a webinar on rootstock breeding will be interesting based on what we discussed. But sure, we can discuss this through emails and elaborate more. Please also note that all attendees will receive probably tomorrow the PDF presentation by Ksenia. And soon also this talk will be available online. Thank you very much.
Thank you very much. Bye-bye.
Thank you. Bye-bye.
Bye-bye.